Hello everyone, and welcome to today's webinar titled Enhanced Rapid Target Identification by NGS for Vaccine Development. This webinar is part of the ongoing coronavirus virtual webinar series. I am Kaylee Bach of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. To learn more, visit thermofisher.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. I now present today's speaker, Cris Kinross. Cris Kinross graduated in Neuroscience from Allegheny College, where she investigated the molecular role of stress and aging on brain resilience. Her focus on NGS development has contributed to the democratization and simplification of NGS technology. For a complete biography on your speaker, please visit the Biography tab at the top of your screen. Cris, you may now begin your presentation.
This is certainly a topic that's near and dear to us because as a company, our mission truly is to enable our customers and the scientists in the world to make the world healthier, cleaner, and safer. At our core, we are all scientists, and this is our purpose. And so we feel like sharing thoughts on this might be helpful for the community. The first stage of vaccine development is discovery. This is where we identify the actual disease. The next generation sequencing, or NGS for short, is typically the primary method used in this stage. Vaccines are developed using specific strains. And so the goal in this stage is to identify each of the pathogenic strains of that disease, understand the rate of evolution of the disease, and identify if it's forming new strains or is fairly stable. So the type of sequencing used to monitor mutations will depend upon the mutational rate of the virus. Coronavirus, for example, mutates fairly slowly, about once a month on average. And so the most efficient way to understand how the virus is spreading or evolving is to use whole genome sequencing, because this allows us to map that entire genome. The next stages of preclinical development and clinical development will be the focus of other case studies in this series. We invite you to review those studies as well, because it will allow you to follow the entire process. Vaccine development is often an iterative process with a sense of urgency. So if we consider one current example, the novel virus COVID-19, the current viral outbreak as of March 2020 timeframe, we can look back to December of 2019 when people in Wuhan, China began to fall ill. That illness has been rapidly sequenced and characterized in nearly every country in the world as of March 2020. An example of the speed of characterization provided by NGS techniques can be seen in Nigeria. On the 27th of February, Nigeria confirmed its first case of COVID-19 at the infectious disease hospital in Yaba in Lagos. On the 1st of March, a clinical specimen was sent to Redeemers University in Nigeria and the Center for Human Virology and Genomics, the Nigerian Institute of Medical Research for Sequencing and Molecular Characterization. The genome was published on GitHub on March 4th. The turnaround time from sample receipt to sequence generation was 3 days. The speed offered by NGS techniques accelerates timelines for vaccine development by providing a clear genomic signature as well as insight into that rate of mutation. So for example, the first Brazilian sequence closely resembles the samples that were sequenced in Germany. But the second Brazilian genome is actually more similar to the genome sequenced in the United States. Comparison of genomic profiles enables study of different viral strains so that we can select the virulent or the most aggressive strains to target for vaccine development. So the Colibri portfolio from Invitrogen at Thermo Fisher Scientific is really based on a simple mission. It's time for NGS library prep to evolve. And one area in which older library prep kits is lacking is in the use of clear liquids and clear tubes without any in-process feedback. At the end of 1 to 2 days of the bench, you find out if you made libraries. This approach wastes time as well as very precious samples that could fail and need to be reprocessed again, assuming that you even have sufficient material remaining. Complete lack of feedback when working with clear liquids and clear tubes is one of those problems which we address here. So you can see a schematic from the manual, which shows how the tracking dyes indicate success. The video shows actual dyes being mixed. You can see where the reagents aren't properly mixed by the distinct red and green layers. You can see proper mixing with the transition to purple. These tracking dyes allow you to save those samples that would have otherwise been lost because you can confirm proper technique by your robot or by the technician at each step. These tracking dyes also allow you to move more quickly with higher success rates compared to older library prep technology because you can visually confirm proper technique at each step. You don't need to go back and mix everything again. You can see immediately if the step was performed accurately and just keep going. So when it's 30 minutes into your library prep protocol, which only takes an hour and a half, how do you know if it's working? So with these Colibri kits, you get the feedback at every step. The other area where Colibri library prep kits are offering improvements is to reduce the need to source reagents and materials from multiple vendors. This has been a big problem with the COVID-19 test kit development in finding supplies from multiple vendors. This library prep kit includes everything you need in a simple SKU. It has adapters, PCR reagents, if that's appropriate, cleanup beads. It even has spare seals. This lets you focus on the science and just keep going. So while multiple sequencing methods for Illumina systems are supported by the Colibri portfolio, in this discussion, we're actually going to focus on two methods, which are whole genome sequencing, which we use to assemble novel genomes, as well as monitor the mutation rate of diseases, but also on 3' mRNA sequencing, because this is used to understand changes in host gene expression, changes in pathogen gene expression, but also how the two interact. So differences in gene expression among people who become seriously ill in response to a disease compared to people who experience only mild symptoms can be really important for vaccine development. So let's start with whole genome sequencing. Here you can see a summary of the various details for DNA that's been physically sheared. This option is available for genomes of all sizes, so it can be used to assemble the genome of a novel virus, but also of the person infected with that virus. For RNA viruses that have been reverse transcribed into double-stranded cDNA, these kits are a big improvement over transposomic prep kits because these kits are really fast. The libraries are generated in about an hour and a half. So we offer options for physically sheared as well as enzymatically sheared DNA, and they both provide the same sensitivity of detection for copy number variants, for variant calling, but from very low samples all the way up to very high samples. You get the entire genome, which is a little bit different from transposomic methods, and it's been thoroughly tested with degraded samples such as FFPE. The DNA kits are extremely consistent, and this is across different users, so it minimizes batch effects, but it also allows you to have consistent performance from low input to high input of DNA. So this consistency is unique to the Colibri kits. And what it means is that you get the same variant calls and the same genomic coverage from a small amount, say from blood, as from larger amounts, what you might get from buccal swabs or from nasal swabs. You aren't limited by the amount of sample you can collect, and therefore you can collect what you have rather than turn away potentially very valuable samples because there wasn't enough DNA in them. Achieving coverage of the entire genome is a really critical aspect of vaccine development, and this just isn't possible from transposomic shearing methods. Transposomic shearing methods rely on near random insertion of transposome complexes throughout the genome. You may have heard of this method referred to as tagmentation. The one drawback of tagmentation is that the ends of the genome can't be sequenced, and this limits the amount of information available from your sequencing data. The biggest advantage of transposomic methods is that they're really fast, typically about an hour and a half. But for the same amount of time, roughly an hour and a half, with the Colibri kits, you actually get the entire genome. So using a sharing and ligation approach, the entire genome in Colibri kits is converted into libraries on Illumina platforms and assembled using standard informatic tools, nothing special needed. Because you get the entire genome, whether it's a small genome similar to a virus or a larger genome such as human or bat, if you're looking into animal reservoirs, you actually get the complete information that you're looking for. The DNA kits have been used already with really badly degraded samples and very low inputs of those degraded samples. Dr. Carla Plavova at Masaryk University and EMBL used these kits to investigate circulating tumor cells in peripheral blood from liquid biopsy research samples. The samples were obtained with consent from people living with chronic lymphocytic leukemia. She compared results from tumor cells against results from buccal swabs that were sequenced on a NextSeq 550 system from Illumina. You can see here the red and blue lines, which reference copy number variation, which were observed from tumor DNA and cell-free DNA using this kit. The results show really high sensitivity of detection from the liquid biopsy research samples at low sequencing depths of roughly 5 to 6x coverage. These kits are extremely sensitive. Dr. Plavova went on to investigate if the Colibri kits were able to detect differences between research samples from patients who were currently undergoing treatment compared to patients who had not yet begun treatment or were not eligible for treatment. This type of test indicates that if the library prep kit has a high enough accuracy to reliably differentiate between different experimental conditions. So a similar example in vaccine development would be to distinguish between strains of a particular virus with that same sensitivity. Dr. Pavlova was able to reliably distinguish between samples from patients who were undergoing treatment compared to samples from patients who were not able to access treatment. The sequencing depth of these libraries was very low. It's only 5 to 6x coverage. And it was also performed on a NextSeq 550 system from Illumina. The results indicate that there's really high GC fidelity, which accurately represents the initial sample in the resulting sequencing data. So this solves this problem of garbage in, garbage out. What you put into the kit, you will actually see in your sequencing data. So we talked about two methods for sequencing for vaccine development. Other big advancement in NGS that's being used in vaccine development right now is 3' mRNA sequencing. This method provides gene expression data from as little as 2 to 5 million reads, and so it can be sequenced quickly and expensively, but also have the sequencing data analyzed very quickly. So when speed is absolutely critical and you need to retain all of the information, these options are great. Gene expression patterns can provide insights into why one group of humans might experience the sniffles when they are inflicted with a novel virus compared to much more serious outcomes, such as hospitalization or even worse. Gene expression data provides new insights when coupled with genomic data from whole genome sequencing to permit development of vaccine subtypes that reduce side effects and boost efficacy, and to be able to do that very quickly. The 3' method works through a series of rather clever blocking oligos, as well as choice of enzymes that result in high coverage of the 3' end of the transcript without covering the rest of the transcript. So what this allows you to do is analyze the full difference in gene expression much more quickly and affordably without investing in very detailed and expensive analysis pipelines. The low number of reads for 3' sequencing, which can be as low as 2 million reads per sample, gives you flexibility in choice of instrument and speed. So you can choose to run many small batches on a low throughput system, such as the mini-seq system from Illumina, or you can batch up a large amount of samples and run them all together very inexpensively on a NovaSeq system, also from Illumina. The data quality across the systems is so similar that your projects become a lot more flexible compared to a method such as whole transcriptome sequencing, because other methods, such as whole transcriptome sequencing, you would need 30 to 60 million reads per sample to get the same information. And so this starts to limit your choices. Flexibility using the 3' system also gives you flexibility in geography. So you can run tests locally throughout the region, and you don't have to rely on large sequencing centers to process your samples. You can actually now generate data in the field near the source of the sample very quickly and very consistently. The 3' method is already being used to study infectious disease. You can see here data that was presented by Dr. Nicholas Mayness of Tulane University for Zika samples from primates. RNA sequencing previously was too cost prohibitive and frankly just daunting in terms of data analysis. And so Dr. Mayness's first comparison was against the gene expression results he previously had detected by nanostring panels. This isn't a perfect comparison between groups because he tested pregnant females against males. And so you would expect to see differences in gene expression. But what he was looking for was depth of information. And he could see in this initial screening that he received a far richer data set from 3' NGS sequencing compared to a nanostring option. So because he was with the initial result, he went on to test it more deeply. His next test was to explore ease of data interpretation. So using a very simple in-house pipeline, he was able to visualize metabolic programming, but also to determine enriched gene sets. This was particularly exciting for him because the Colibri gene expression data sets are far richer compared to what he had previously been able to achieve with nanostring, and they were actually at a lower cost. And so he announced a few weeks ago that he intends to use the Colibri 3' mRNA kits to begin COVID-19 viral studies immediately now that NIH funding has been released in the United States. All data sets for the Colibri RNA portfolio can be analyzed and interpreted using the Genialis expression software. Their software contains pre-built analysis pipelines for whole transcriptome sequencing, mRNA sequencing, and 3' mRNA sequencing from the Colibri portfolio. It's designed for biologists rather than informaticians. If you'd like to get a demo of this software, we encourage you to contact Genialis at the email address shown here. The NGS informatics community has been so good at working together, and they've rapidly released tools to access coronavirus genomes from each region. These databases are available for free to vaccine developers and researchers of all flavors. And so while Thermo Fisher Scientific doesn't endorse or recommend any specific analysis package, a simple Google search will yield extremely valuable options, and this will help you to accelerate and simplify your informatics because you won't need to build your own database. You can access what the community is already built together. We support the NGS community with a rather generous sampling program. You can visit the URL shown here to request a sample or contact your Thermo Fisher Scientific representative. We'd love to be able to help you. So with that, I thank you for your
Thank you, Cris Kinross, for your informative presentation. As a reminder, our speaker will follow up with any questions you have submitted via email following this webinar. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and their interesting questions. Questions we do not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand through the end of this year, 2020. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.